Hello, everyone, again. Welcome to the last session of the Congress. So the first speaker is Alvaro Benito, and the title of the presentation is Live Myco Restore Use of Natural Myco Control Products and Native Fungal Species for Reduced Species Phantom Presence in Forest. Okay. Okay.
Good morning, everybody. I'm going to speak about the LIFE project, Myco Restore, that it's ongoing. It's a project that is ongoing, so I'm going to speak about the work that we are doing right now and the different results that we already have. First of all, to speak about a little speech about the LIFE. The LIFE is a LIFE project that is involved different organizations of European countries, mainly from Italy, Portugal, and Spain. And it's focused in three different parts: mycoeconomy, mycoforestry, and mycocontrol. We at UBA, we work in the part of mycocontrol, mainly with the mycopesticides, mycorrhizal biocontrol, laboratory trials, and non-toxic pest management. The work that we do are all in the region of Salamanca and in 4 different villages. In La Medilla with Quercus ilex, in Linares with Castanea sativa, in Cubo Don Sancho with Quercus pyrenaica, and in Valdelosa with Quercus suber. Our work it can be divided in 3 different parts. First of all, we want to identify the fungal biodiversity. We do this thing through culturomics, trying to isolate the different fungi from these plots, and with a metabarcode analysis. After the identification of the fungal biodiversity, we do trial in the laboratory, trying to find antifungal species through antagonist test, and trying to find an entomopathogenic fungi to control different insects that are in the plot that are causing damage in the area. And to end the project, we are going to do trials using mycopesticides, all with native fungi. Well, first of all, we have to identify, as I said, we have to identify all the fungal biodiversity. So first of all, we have to do a sampling. We go to the different areas and we collect field data, fresh tissue, and soil sampling. With this fresh tissue, go to with the first task, that is trying to isolate as much fungi as we can with a culturomic assay. And we take the samples that we collect in the sampling, branches, leaves, bark, and wood, and we take little pieces. We sterilize the surface of these pieces with alcohol and bleach, and we put in plates with PDI, streptomycin, and ampicillin, that are 2 antibiotics that avoid the growing of bacteria. Then we put at 23 degrees to permit this fungi to grow, and then with all the fungi that grow in these plates, we try to re-isolate to have a pure plate with the different strains. And this is an example of the work. We put the different pieces of, for example, leaves in a plate. We have a lot of fungi in one plate, and then we try to take each fungi and put it in individual plates. Well, this is the one part of the identify of the fungal biodiversity. The other part is the metabarcode analysis with the branches, leaves, bark, and cork that we collect in the field. We try to extract all the fungal DNA and go to a sequencing. With this analysis, we can know which fungi do we have, with how many fungi we have, and what are the function of these fungi. With this metabarcode analysis, we detected 892 fungi. The main interest of the principal fungi that we detected was these ones, for our project, obviously, is Beauveria species, Trichoderma species, and Cordyceps species. There are 2 of these, Beauveria and Cordyceps, that are entomopathogenic, these fungi affect the insects, and we have trichoderma that is an antifungal species. After the identify of all the fungal biodiversity, we go to the next step, that is the trials in the laboratory. With all the collection that we have from the culturomics, we take this fungi and we put in the same plate with a pathogen. In this case, it's Cryphonectria, that is a disease of the Castanea sativa, of the chestnut. We put in the same plate, and if we find at the initial step, we put four fungi from the collection with the Cryphonectria, and if we see some antagonist effect, we pass to the next step, to the final antagonist step, in which we put one fungi in front of the pathogen. And then if we see that we have an antagonist effect, we send to sequence to know which fungi is. This is one part, the antagonist test, to select fungi that can be used to control a pathogen, and then we have the other part that is to find entomopathogenic fungi to fight against two insects that we have in these plots that are Coraebus undatus and Cerambis wellensi. Both insects attack the wood of the trees, causing damage in the trees. First of all, we have to do an insect trapping to get these insects from the field. After that, we get with all the fungi that we have from the culturomics assay, we take this fungi also to put with the insects to see if they can kill or they can influence the life of the insect. And if we have good result, we pass to the next step and we put the fungi with a plant assay to see if the fungi can grow or not. And then we inoculate in forest if we have good results. The last part of the project is use these fungi to inoculate in the forest. We have a problem in the area. We have two insects of the same that are very similar. Cerambis wellensi and Cerambis cerdo. One is protected by the European Union. So now we are sampling in June and July. We are sampling the area to know if we really have this Cerambis cerdo in the area. And after knowing that, we can make a decision about if we can use the different antifungal species, different entomopathogenic fungal or not. Also, in the beginning of the project, we plan to use commercial fungal entomopathogenic to fight against these insects. But the Junta de Castilla y León only allows us the application of fungi that we isolate from the field with the culturomics. So this marks our work. And that's all. If you want to know more, sorry. Thank you. But if you want to know more about the project, you can go to the website. And if you want to know more about our work on the research group, you can go to the Twitter. And every week we post different things about our investigations. Thank you very much.